In this video, we're going to pick up where we left off talking about the about the organelles that you can find in a cell. And so we'll get started with mitochondria. Mitochondria, so if we're thinking about the cell as a town, perhaps, if we have that as our analogy, the mitochondria are like the powerhouse in the town. It's a place where power is generated. And the work of the mitochondria is to convert the chemical energy of glucose into the chemical energy that we can store and store for a very brief time in the chemical adenosine triphosphate, also abbreviated as ATP for adenosine triphosphate. And we do this work by essentially burning glucose, by reacting glucose with oxygen in order to release the energy that we find in glucose, and then that energy that gets released is converted into another kind of chemical energy by making ATP. So the job of the mitochondria is to produce ATP, and it does it using glucose and oxygen. And along the way, it produces a side product, a waste material, carbon dioxide. And so that's why we have to transport oxygen around the body. It's to get that oxygen to the mitochondria of all of our cells to allow them to be able to burn the glucose. And then we produce all this waste carbon dioxide, and we have to carry that around to get it back to the lungs so that we can breathe it out. The mitochondrion is a double membrane organelle. So it's got an outer membrane, that's this here, and then this highly folded inner membrane that loops around and is very complex and very folded up. The folds in here on the inner surface are referred to as cristae. AE always says E, so you don't say cristae, but it's just cristae. And all that folded up membrane provides lots and lots of surface area. Anytime we see that a membrane is folded like this, the first suspicion should be that, we have a process where we need some more surface area for something to occur. And in this case, what's occurring is that there are dotted all over this membrane lots of different enzymes that do the different processes of this converting of glucose into ATP energy. And so all that folding gives us lots of surface area so that we can have more enzymes packed inside of this mitochondrion, giving us the ability to make lots and lots of ATP. And the more a cell uses energy, then the more ATP we'll generally see associated with that cell. Alright, so that's the mitochondrion, responsible for producing usable chemical energy for the cell. We also have organelles that are responsible for breaking up and destroying materials inside of the cell. We can think of them as the garbage disposal or the recycling center for the cell. And they're called lysosomes. Lys means to break up. Somes, that soma means body. So this is a body inside of the cell that has the capacity to digest and break up stuff. So lysosomes are vesicles. A vesicle is just a membrane-bound sac that is filled with all kinds of enzymes that are very good at, whose function is to digest or to lyse lots and lots of different molecules. And so it has the capacity to break up any of the material that gets brought into the cell as food so that we can use so that the cell can use the nutrients that are then digested out of the food materials that come in. Or if it's a white blood cell that's going around cleaning things up, say maybe finding bacteria and then absorbing them, bringing them into the white blood cell inside of a little package, then that little package of bacteria and nastiness can be fused with a lysosome, and then the bacteria get destroyed. So lots of these enzymes are they function best if they are in an acidic environment, and so they're referred to generally as acid hydrolases because they work best in acids, and their job is to digest or to lyse chemicals by breaking up the biological molecules, the proteins, the carbohydrates, whatever it is that's being brought in, breaking it up by using water as the means of breaking it up. They're also used in a process known as autolysis. Auto means self, lysis means, as we've already said, breaking up. So it's sort of like a suicide sac inside the cell, and if the cell gets the word that, hey, we need to die, then it can activate the lysosomes that are inside of it. They open up and the enzymes make their way through the cell, digesting everything and eventually killing that cell. And this can be really important if we think about, say, the development of a little baby, and we're thinking about the hand. The hand starts as this kind of paddle shape, but that's not what a hand looks like. And so the cells that are in between the fingers to be, oh gosh, this poor little baby, it's got some really bad looking fingers, but you get the idea. These cells all have to die in order for the fingers to be separated from each other. And so they undergo autolysis or autolysis, and as a result, the paddle then can look more like oh gosh, I still can't draw a hand, but you get what I'm saying, hopefully. Alright, so the next of the organelles are called peroxisomes. These two are vesicles, that is, they're just little membrane-bounded organelles that are filled with a kind of enzyme called a peroxidase. Peroxides are very dangerous to a cell, and a lot of the cellular reactions that occur inside a cell, a lot of the metabolic reactions, produce dangerous peroxides like hydrogen peroxide, the same kind of material that we would use to clean a cut. And you pour that onto a cut onto a cut on your hand or wherever, and it bubbles very vigorously. And so it does the same thing could happen inside of a cell. You could start a reaction that would be very dangerous to the cell. And so there is a enzyme called catalase in these peroxisomes that will break down this really dangerous peroxide, like hydrogen peroxide. So we use it in we use these peroxidases to produce bile acids that are used to digest fat. They can use it to break down lipids, to degrade really strange biochemicals, and to detoxify alcohol. So a lot of different things get digested in peroxisomes, and they are especially abundant in the liver and the kidney cells, because that's where we do most of this this breaking down of these chemicals that along the way will produce these peroxides. The centrosome is then the next of the organelles that we're going to talk about. Centrosomes are these two structures called centrioles. Centrioles are like a barrel-shaped structure, so they're round, and they're made of these clusters of microtubules, special protein fibers that are arranged in a barrel shape. So the way that the centrioles are oriented to each other, though, when I draw them, I usually draw them like this. So that kind of gives you an idea. Here's one barrel, and here's the other barrel, and they will be oriented at right angles to each other. And generally, during a lot of the life of a cell, there's only one centrosome, one of these set pairs, associated somewhere near the nucleus. But during cell reproduction, the centrioles will reproduce so that there's now two sets of these centrosomes, and they will migrate to opposite ends of the cells, to opposite sides, and they become an anchor for this really complex series of microtubules that will span out across the cell and will attach to the chromosomes and help to pull apart the duplicated chromosomes. So we see over time that the chromosomes, as they're being pulled apart, will get further and further apart. They get drawn back to the centrosome that's at their pole until all of the chromosomes are here at one pole or the other, and then the cell will divide. So that's all involved in separating the chromosomes so that each of the new daughter cells will have its own set of chromosomes in it. We also see centrioles as basal bodies. These are anchor points at the base of flagella and cilia, the movable parts of the cell. So centrioles, when they're together as the centrosome, they're going to be anchor points in the cell that we can tug or twist something associated with the cell. So we can tug apart the chromosomes, or we can twist a flagellum, or make a cilia flap. Okay, so that's the centrosomes. Cilia, these are short extensions of the cell membrane. So if this is the membrane of the cell, then it has these short extensions, and inside of each of the extensions is a set of microtubules that give it some structure, and these microtubules will wave. They can wave one way, and then they push material the opposite direction. So by waving, they if we were to look at it, they would go back, and then we would see them push forward, so that whatever material is, what fluid is around the cell, will then be pushed in this direction, and then they can kind of reset and then push again, so that they can push substances like mucus across the cell's surface, so across the surface of a set of cells that are all ciliated. And so we can find cilia like this in the respiratory system. So if we were to look down the trachea into the bronchi and then deep into the lungs, we would see that all the surfaces of these tubes bringing air into the lungs are covered up in cilia. And the cilia, and embedded in there within the cilia cells, the cells that have these cilia surfaces, are other cells that will squirt mucus up to the surface. So as we breathe air in, if there's dust particles in the air, then the mucus that lays across the top of the cilia here will collect those dust particles, and then we can wave the dust particles up out of the lungs, back into the bronchi, back up the trachea, and then when we get into the back of the throat, there at the pharynx, one little swallow, and all of that mucus with whatever dust and bacteria and viruses that we breathed in all go down into the stomach, where the acids and the enzymes down there will digest the materials, will render them non-harmful, because the bacteria won't survive, and we can take care of any material that gets into the lungs that way. Now, one of the downside about, say, doing something like smoking is that one effect of nicotine is to paralyze the cilia for a brief time, for a half hour or so. So while a person is smoking, they're bringing in all these smoke particles, and they are also paralyzing the cilia, preventing the lungs from clearing themselves. And so it takes quite a while for a person who quits smoking to get their lungs re-cleared again, because there's been a lot of buildup of stuff that gets in there because of this paralyzing effect that nicotine has on cilia. And here we can see cilia sliced in cross-section, and you can make out the pairs of microtubules that are arranged in a circle around a central pair. And as those microtubules rotate around a basal body, a centriole at the bottom of each one, then we can get this wave-like motion that pushes the mucus, or say fluids of the fluids in the kidney tubules get moved along by cilia. The fluids in the ovarian tubes, the fallopian tubes, I'm sorry, I said ovarian tubes, I meant uterine tubes, is also moved along by cilia, because there's cilia that line the uterine tubes, also known as the fallopian tubes. Okay, so that's the cilia. They move a fluid across the surface of a cell. Flagella are also built in a similar way. There's only one, but the flagella are much, much longer. And there's only one cell in the body that is flagellated, and that's one of these sperm cells. So here's the head, there's a little mid-piece behind the head, and then the rest of this is a flagellum. And it allows a sperm cell to swim through a watery medium, so a mucousy, watery medium. And like I say, constructed similar to a cilia, but much, much, much longer than cilia are. Alright, so we're getting now to the end here of the organelles that we find associated with a cell. And so the next set of organelles we're going to the next set of inclusions in organelles are the cytoskeletal elements. So the cytoskeleton of a cell is a network, a mesh of protein filaments and microtubules that support and maintain the cell shape. So if we're looking inside the cell here, here's the cell membrane, and inside of the cell are this complex arrangement of microtubules and these long microfilaments here that help to provide structural support for the cell. They provide this network, almost like a set of train tracks that we can use to drag materials from one location to another in the cell as well. So microfilaments, they are composed of a specific kind of protein. It's like a big long chain of these specific kinds of proteins called actin fibers. And we're going to discover more about actin fibers when we start talking about muscle cells, because they are a big part of the process of movement in a muscle cell. And then there are also these long, flexible, hollow tubes called microtubules. They're built of a protein called tubulin, and we see them in building centrioles. So centrioles have a lot of microtubules in them, as well as in the cilia, supporting and causing the movement of cilia and flagella as well. And there are lots of other kinds of microfilaments. We have intermediate tubules. We have there's just a lot, intermediate filaments. I'm sorry, not intermediate tubules, but intermediate filaments and other kinds of microfilaments and microtubules inside of the cell that give the cell its structure and shape. Okay, so the last of the inclusions inside of a cell include then the nucleus. I think of this as sort of the boss of this factory, or the mayor of the town. The nucleus has access to all the blueprints required to build and to maintain the cell. It's a very large structure. It's spherical, and it's found generally toward the center of the cell. And like the mitochondria, it is a double membrane organelle. But rather than being highly folded, the double membrane, the second membrane, is just inside of the outer membrane. And it houses all of the genetic information, the DNA of the cell. And it's responsible for controlling the cell's activities and determining when the cell will divide. Now, most cells have a single nucleus, but there are a couple of groups of cells in the body that can have multiple nuclei. So skeletal muscle cells have lots of nuclei. Some liver cells will have nuclei, and then there are others as well. And then there are even some cells that have no nucleus at all, because red blood cells, once they mature, will eject their nucleus out of the cell, and it's gone. And so a mature red blood cell is actually unable to repair itself because it has sent away the blueprints, so that it has nothing but it's just stuffed with hemoglobin. There's just no room for even the nucleus in a mature red blood cell. This double membrane has pores in it, little openings that penetrate and will allow materials to come in and out of the nucleus, but will not allow the DNA to escape. Okay, so if we look closely at the nucleus and some of the structures there, there's the outer nuclear envelope, sometimes called the nuclear membrane. Either one is a very appropriate term. It's a double membrane, as we said before, so it's got this phospholipid bilayer with another phospholipid bilayer just outside of it. There are the pores, the nuclear pores, that allow materials to go in and out of the cell, and they are very strict about what can get in and what can get out. It's filled with a nucleoplasm, so there's cytoplasm inside the cell. Inside the nucleus is nucleoplasm, and there may be one or more of these darkly staining spherical bodies inside of the nucleus that are referred to as the nucleolus. It's very tempting to want to call that the nucleolus. It's the nucleolus. So nucleoli, that's the plural for nucleolus. These are small, dark staining bodies inside the nucleus, and they're built of proteins and of a kind of material called RNA. And their job is to actually build the ribosomes. So they produce ribosome parts. So the nucleolus is here. It's tempting to think that the nucleolus is where we find the DNA, but that's not the case. It's just there to produce ribosomes, and so if it's a cell that has to make a whole lot of proteins, then there may be lots of nucleoli inside the nucleus. The DNA is referred to as chromatin when the cell is in the middle of the cell cycle, when it's just doing the function, the work of the cell. And chromatin is a long, strung out strands of DNA. And they are not visible under a light microscope. And they're associated with these proteins called histones that are kind of like little spools. So they'll be wrapped up around a histone, and then there'll be a length and another histone, and they wrap up around it. Prior to beginning the process of cell division, the chromatin will coil up very tightly. So it coils, and the coils coil, and then the coiled coils will coil again. So it supercoils into a very tight bundle, and then those supercoils become visible because they're large enough and dense enough that we can see them, and we refer to them as the chromosomes. So chromatin is DNA kind of stretched out. Chromosomes are the DNA condensed so that they're very compact and they become visible at that point. Okay, so there we have it. All of the organelles that you find inside the cell. And so be sure to be able to explain the function and the structure and the location in the cell for each of these organelles, so that that will, those being able to answer those questions will help you to know all the material that you need to know about each of these organelles. All right, as always, any questions, give me a call. Or, better yet, send me an email.